Okay. This is an image from a textbook. It is an electron microscopical image and shows a lymphocyte. Yeah, so a human lymphocyte which has been stained with a specific staining with a residuum red staining, which is required to see also the sugar residues on the cell surface. And the sugar residues on the cell surface is the glycocalyx. Yeah, you see here as the lymphocytes have a very big nucleus. It has a small, relative small cytoplasm, and then a very big nucleus, a very small cytoplasm, and then it has a small rim, which is labeled here in black, and this is the glycocalyx. So it's a tree of sugars which is on top of the proteins and the lipids, and which is important for regulation of interaction, cellular interactions, and also important for interaction with the immune system. The neurons are very rich in the sialic acid glycocalyx. So if we look into the brain, there's a huge amount of sialic acid present, and we believe that it's important to keep the immune system in check in the central nervous system. So you have here the glycocalyx with a sugar tree, and on top, this is labeled here in red, it's the sialic acid. So the sialic acid is found on lipids, and the lipids in the brain are normally the gangliosides. Yeah? So the gangliosides have a lipid tail, and then they have a short sugar chain, and then normally they have also sialic acid attached. Or it's found in proteins. Yeah? So we have several proteins. So most of the proteins on the cell surface, but also proteins which are released from the cells, also in the serum, the proteins which we have are glycosylated, and normally on top they have the sialic acid. So the sialic acid is the silylation, and it's on a specific position, on a position ready to interact mainly with the immune system. And this is giving a signal for an intact situation, an intact cell, an intact protein. Yeah? So if proteins in our serum are aging, they lose their sialic acid due to oxidative damage or due to a specific enzyme, and then our system will recognize it and will say, yes, this is an aged protein, and we remove it. And the same is with the case with the cells. So sialic acid on top is a signal for intact. So telling the immune system, don't touch, don't remove it, keep it. It's an intact cell as a signal system. So sialic acid. So where is sialic acid coming from? It's normally here produced inside, or it's taken up, either it's taken up from outside, from other glycocalyx sugar proteins, and then it's removed by neuromidases, and then it's activated in the nucleus by an CTPase, and then you have CMP sialic acid, and the CMP sialic acid is attached to the lipids and the proteins by specific sialyltransferases, and then they are transported on the cell surface. So on the cell surface, they are ready for interaction, but there they can either be removed by oxidative damage or by specific enzymes again, or by removing the whole protein together with sialic acid, and then they can be taken up. On the other side, the cell is able to produce its own sialic acid, and this is coming from the glycolysis. So you have precursors of the glycolysis, and then by certain enzymes you can produce in all cells N-acetylneurominic acid. This is the sialic acid forms which we have in humans. So some years ago, we did a very simple in vitro experiments, and this was a very crucial experiment for us because it was telling us, yes, sialic acid is important in the central nervous system. So we cultured neurons, and normally neurons in culture have a glycocalyx, they have sialic acid on top. And now we treated these cells with a neuramidase, so with specific enzyme to remove only the sialic acid. So if you remove the sialic acid on a cultured neuron, there was nothing happening. So the neuron was still intact, it looks fine, it was surviving. However, if we added microglia, then the microglia immediately started to remove the neurons. So if we added microglia to a neuron which has an intact sialic acid glycocalyx, nothing was happening. The neurons even looked more healthy. Yeah? So the microglia supplied trophic support even for the neurons. However, if the neurons have no sialic acid, within a few hours, they were binding and they were removing the neural processes and the neurons. So the neuronal silyation was preventing this immune attack and this immune-mediated removal, and we found out that the complement system was involved. So complement was binding if there is no sialic acid, and then the complement cascade was activated. And then this complement cascade allowed the complement receptor CR3 of the microglia to phagocytose away the neurons. And this was the case in the mouse as well in the human. So it was a key experiment for us. And then our research completely shifted into this topic about the sialation and the protection of the neurons by the sialylated glucocalyx. So the question then we were trying to ask is first, how does sialation prevent complement activation? Yeah? So on one side we showed, yes, if you have lost sialic acid, we have binding of complement C1q and we have a complement activation cascade. But this was also known from literature. Yeah? So in old days, it was shown that sialation prevented complement activation by a specific complement inhibitor, which is called complement factor H. But if you looked also in the old literature, polysialic acid, so a chain of several sialic acid as a polymer, was also able to prevent complement-mediated lysis of bacteria, which were having this polysialic acid on the cell surface. So as I mentioned, there have been on one side, again here we have a schematic drawing, we have here the glucocalyx, the sialation glucocalyx, and then as an output we have complement activation. So complement activation is normally a cascade, and this can be triggered by direct binding of complement factor C3 to lesion cell surface. But the factor H is an inhibitor, which is labeled here with factor H. And this factor H binds to sialic acid, binds to complement C3, and prevents a complement activation. So this was a known concept, so we didn't follow it up. This is well described in the literature. What we did is, from the literature, at least indirectly, it was also known that polysialic acid, so a chain of several polysia molecule, can be attached also to the glucocalyx, but can be also released, also prevented complement activation. And here the mechanism was completely unclear. So we were studying how polysialic acid prevent complement activation in different assay systems and all cultured systems. So first we were using an ELISA to analyze the complement activation, and this is an assay system, ELISA system, which you can buy commercially. It's called the VSLAB assay system. So the system is set up that you have a ELISA blade, which is coated with lipopolysaccharides. So it's a blade-bound lipopolysaccharide. So normally then you add just human serum. You add human serum, which is completely intact, yeah, so which is not heat treated. If you heat treated it, and which we do very often for cultured cell, then we inactivate by the heat treatment the complement. So we take complement intact human serum, and if you add this to this culture blade, the lipopolysaccharide will induce a complement cascade, an alternative complement activity, an alternate complement cascade. And the final readout of this complement cascade is the formation of a terminal complex, complement complex, a TCC, which contains C9 and C5b. So, and this is the situation, the readout, the signal, if you have normal human serum to the blade. If you now added monosialic acid, so a single sialic acid molecule, to this assay tube, nothing was happening. If you added now oligosialic acid, so these are 3 sialic acid in a chain, so it's a polymer with 3 sialic acid, nothing was changing. The same was the case with 5 sialic acid in a chain, nothing was changing with the complement activation. However, if we have a chain length of about 20, yeah, so 20 sialic acid molecules in a row as a polymer, then we saw a very strong inhibition of this alternative complement activity. And the same was visible if we added very long chain polysialic acid, which you normally find on specific bacteria, which are really pathogenic for humans. Yeah. So there we also partially and with a high variability saw a complement inhibition. So we were saying, yes, with this assay, we were able to show that low molecular weight polysialic acid, how we call it, so polysialic acid with a chain length of 20, was able to inhibit the complement activation. So then if you have alternative complement activation, you immediately will think about properdine. Yeah, so properdine is an initiator and a stabilizer of the alternative complement cascade. So it's the only known activator of this alternative complement cascade. So it can bind to lesion cell surface, and then it can recruit the C3 convertase and can lead to an alternative complement activation. And as a second, it can stabilize this complement C3 convertase, and by this it can amplify the complement signal. Yeah, so it's a classical activator and amplifier of the alternative complement activation. So we were asking, so what is now happening with properdine? Yeah, so we were taking cells, put them in culture, and these have been, for example, a neuroblastoma line, but we also were taking a mouse hepatoma line. So it was working out with several different cell cultures. So normally, if you add now to these cultured cells, and this is shown here in the second image on the top, a normal human serum, and we have lesioned these cells before. Yeah? So they have been under starvation, so they were not getting enough feeding material. And then if they are under starvation, they are suffering and they have a changed cell membrane. And this is normally recognized by the complement system as a lesion cell membrane. So if we now added normal human serum, the complement system is activated, properdine is binding, and these are the white spots which have been then popping up on this lesion cell. So we had properdine binding as an activator of the alternative complement pathway on this lesion cell. And this was inhibited if we added polysialic acid, so 8 micromolar polysialic acid, but also higher concentration, and this is shown here, inhibited this binding of properdine to the lesion cell membrane. So we can say now, low molecular weight polysialic acid, a chain length of about 20, inhibited the binding of properdine to this lesion cell surface. And this is again published in this recent paper in scientific reports. So we were also going one step further. Okay, we were asking, where do polysialic acid, or to what molecule it is binding? Yeah, and again, this is a very simple binding assay. So again, a ELISA plate where we coated different factors, factor B of the complement cascade, factor H, which is known to bind to sialic acid, but not to polysialic acid. Factor H is binding to 2.3 linked to another linkage of the sialic acid. And then we bound properdine to the plate and the different components of the complement C3 convertase. And what we can see, only polysialic acid was only binding to properdine, but not to the other complement factors. So we can conclude low molecular weight polysialic acid specifically binds to properdine, but not to any other complement factors. So the concept which we now have is that, yes, polysialic acid binds to this properdine, which is the activator. And by binding to this activator, it's neutralizing its activity. So properdine no longer can amplify the C3 convertase, or properdine now cannot be targeted to the lesion cell membrane and cannot initiate the alternative complement pathway. So I would like to summarize this experimental part of our data. Yeah, polysialic acid, not the short ones. Yeah, you see, you need a certain chain length, a chain length of 20, but it's working up to a chain length of 40 or 50, inhibits the alternative complement activation. A polysialic acid inhibits properdine opsonization. Opsonization means the binding to a lesion cell surface. And we also can show polysialic acid binds directly to properdine. So we were coming one step closer to the mechanism, how polysialic acid is interfering with with complement activation. So we can now say silyation prevents complement activation by 2 pathways. The one is described in literature since 20 or 30 years. It's factor H. Factor H is an inhibitory of the complement cascade. It's also inhibiting C3 convertase. And factor H has been shown 3 years or 4 years ago that it's specifically binding to alpha 2.3 linked sialylated oligosaccharides. Now we have binding of polysialic acid to propatin, and there we have a different linkage. So the sialic acid molecules are linked in a certain conformation, and this is different for polysialic acid to most of the other sialylated glucocalyx molecules. And polysialic acid is 2,8 linked, and therefore we think that there might be specificity to interaction with propatin. So this means that we have elucidated one mechanism, the sialation prevents complement activation, particularly the polysialic acid. So now we come to the second, what we observed. We observed that the microglia is phagocytosing away the neurons which have no sialic acid on the surface. And we also know that the microglia are activated if there is no sialic acid on the surface. So the question is, how does sialation prevent microglial activation? And there we contributed some research. But if you look in literature, there are many people which are working on this field, which is an exploding field because it has a lot of human relevance, in particular for neurodegenerative diseases. And there we come to receptors which recognize sialic acid. So they don't recognize one single sialic acid molecules. They normally recognize the sialylation path. So they at least recognize free sugars. So they recognize the linkage of the sialic acid to the underlying sugars, and they recognize also normally 2 or 3 sugars, carbohydrate molecules, monosaccharides below the sialic acid. By the Cyclac family, which is called sialic acid binding immunoglobulin-like lactides, we have a huge difference in species. And I'm showing here only the Cyclac family, which has diverged in the different species. And this is called the CD33 related, the Cyclacs, which are the most interesting for us because they normally have an inhibitory signaling. So they are responsible for controlling the immune system. And you see already that the labeling is quite different. In mouse, they have Cyclac EFG, so they have alphabetic numbering mainly. And in humans, they have just numeric numbering. So already the numbering is different. And this means also that you cannot make a clear homology between a human and a mouse Cyclac. So the mouse Cyclac, Cyclac3, CD33, has the same name as the human's CD33, but it has a different signaling cascade. The mouse one has a charged residue, which can also lead activatory signaling, while the humans is only inhibitory signaling. And they have also a different ligand binding property. So there are always no clear homologs between the mouse and the humans. The second is that this gene family exploded in humans, and in particular in human primates. So if you look to other highly developed species, they have even less, and many of the Cyclacs are even human specific. Some people even say that the human species was defined by these Cyclacs and the glycobiology and diverged out of the primates, on the non-human primates, by becoming more specific. And they were developed all by gene duplication. So they were coming very late in evolution and have a lot of gene duplications. And then there were some Cyclacs popping up like Cyclac 16. They even are not unique and diverged in the human population. So several humans have an intact Cyclac genes, about 30% or 40% of the population, and most of us have only a pseudogene. So they have a non-functional Cyclac 16 gene. So it's even diverged in these expression profiles in in different human populations. I think what is more interesting for us as a neuroscientist or neuroimmunologist is that there is a close linkage to Alzheimer's disease and or dementia. I'm always saying that it's not fully clear whether it's only Alzheimer's disease or dementia, because if you look to these large genetic publications with huge numbers of patients, 100,000 or 300,000 patients, you will realize that many of these patients are not clearly characterized in respect to the disease cause. And therefore, I prefer always to say that this is linked to dementia. Also, the people normally publish it as a linkage to Alzheimer's disease. This is known since many years for CYCLAC3, CD33. There is a clear genetic linkage to dementia and Alzheimer's disease. It was becoming very recently also clear for CYCLAC11 in this Belenguez et al. Paper. And they were saying, yes, the CYCLAC11 is even the most important gene of the microglial genes in respect to Alzheimer's disease. So people believe that the genetic risk of Alzheimer's disease is not to 70–80% related to a certain innate immune pathway. And if you now categorize and analyze the pathway, you would say the highest priority of these genes is CYCLAC11 on top. Yeah? So as the receptor regulating this innate immune pathway in the microglia. Interesting is that they published only CYCLAC11, but if you carefully look in the original data, which are in the supplement of this Belenguez et al. Paper, you will realize it's not only CYCLAC11, which is highly linked to dementia, Alzheimer's disease, it's also CYCLAC16. They haven't mentioned this in the publication, to my opinion, because they do not know whether it's the intact or the pseudogene, which is linked to dementia, Alzheimer's disease. And therefore, they just add it as a supplementary data and have not discussed it, because it's very difficult to write something about a gene which is linked where you even do not know whether it's an intact or a pseudogene, a non-functional gene, which is linked to this disease. So apparently we have a high relevance of the CYCLACs for neurodegenerative diseases, possibly for aging and dementia, because we have a clear association of these diseases with the age. Yeah? So this can be also the reason for this linkage and the risk factor. So now we come to the mechanism. How does silyation prevent microglial activation? And this is again a schematic drawing. We have here the neurons, which have the glucocalyx on top of the proteins and also on the lipids. Yeah? On top there is normally sialic acid, and the CYCLACs are bound to recognize. As I mentioned, they recognize not only sialic acid, they normally recognize these free sugars, yeah, or a linkage or a stretch of sugar. So they can also recognize polysialic acid, for example, by the CYCLAC receptors. The CYCLAC receptors can do it in trans, but some of them do it also in cis on their own cell surface, and they mainly have inhibitory signaling. Yeah? Immune receptor tyrosine-based inhibitory motif, which are very similar to this T-cell receptor signaling. So in the T-cell receptor signaling, we have also of the lymphocytes, of the CD4, CD8 positive T cells, we have also activatory immune receptor tyrosine-based motifs, but also we have inhibitory motif. But these are microglia, macrophages, so a different cell type. So normally the inhibitory signaling is only counterbalancing activatory signaling. Yeah? So here is again the intact situation where we have this inhibitory signaling. Inhibitory signaling normally is not doing any harm. It's just there, but it's becoming important if you have an activatory signaling. Yeah? So it's a counterbalancing system, this inhibitory signaling. The activatory signaling is also well known. It's coming from TREM2, which is an Alzheimer's disease gene, yeah, which is again highly linked to Alzheimer's disease, which can bind amyloid beta aggregates, which can bind phospholipids, which are popping out on the cell surface if you have lesion cell. And this signaling can lead to activation of the microglia. Activation means that they increase phagocytosis, they clear off the cells, they remove synapses. You will have an oxidative burst. Yeah? The oxidative radicals then are freely diffusing and are damaging our brain. You have inflammation. You can get migration and proliferation also of this microglia. And again, this activatory signaling is shut down by this inhibitory signaling. So meaning that all these receptors cluster together on a certain spot on the cell, and the net outcome of this signaling is deciding what is the cell doing. The other activatory receptor is the complement receptor CF3, which can signaling via thyro-BP like the TREM2, and it's recognizing the complement cascade, which apparently, as I mentioned, is active as soon as we have lost the sialic acid. So this is the schematic and the basics behind this inhibitory function of the Ciclex in the system. But I always wanted to mention this inhibitory signaling is not always inhibitory. So this IT motif can do also some active parts, and this is, for example, endocytosis. So if this receptor is recognizing a soluble sialic acid structure, it's normally taking it up by endocytosis. Yeah? So the endocytosis as a homeostatic function in the microglia is even activated and increased by this inhibitory signaling cascade. Only the pathologic signaling is completely shut down by the Ciclex receptors, by the sialic acid immunoglobulin binding receptors. So we can summarize, Ciclex receptors are highly diverged, at least the CD33 related family in mammals, with a human-specific Ciclex. And many people believe that the increased lifespan of the higher mammals, like the humans, is due to this inhibitory function of this Ciclex receptor. And if you knock out the main inhibitory Ciclex receptor in the mouse, you get gray hair, you get dementia, you get premature aging, and you have a reduced lifespan due to oxidative burst everywhere in the organism. They are detected on transcription level in microglia, and many microglia express several Ciclex receptors. They counteract the activatory, immunotyrosine activatory motif signaling in the microglia, for example, the phagocytosis and the oxidative burst, but they still allow and activate endocytosis. So meaning that we conclude sialation prevents pathogenic microglial activation via the Ciclex, but keeps intact homeostatic activity of this microglial receptor. So now I come to the next part. So we were also asking the question, what happens if we have insufficient sialation? So I mentioned that the cell can produce sialic acid, and they normally continuously do it. They replace the lost sialic acid continuously. We have even a time period where we have insufficient endogenous production, and this is also the case in humans. So if we are postnatal, so after our birth, we have normally mother milk. So we have breastfeeding. And the mother milk is supplying a lot of sialic acid by oligosaccharides, human milk oligosaccharides. And these human milk oligosaccharides are needed in this critical postnatal period they go into our circulation of these newborns and also enter into the brain and will help to build up this huge amount of sialation in the brain. So you need this early breastfeeding, you need the sialic acid from the mother's milk for getting full intelligence in humans by adding this sialation. So we have time periods where we have insufficient sialation, and we need exogenous sources in a very early postnatal period. But in our concept, it could be that it's also the case in aging. So in our understanding, apparently, we have also insufficient endogenous sialation in aging. And we would postulate that also in aging, sialation supplementation, you need not monosialic acid, you need complex sugars to do the job, might be beneficial to prevent the age-related inflammation and diseases. So what is happening if we have now reduced sialation and we mimic this in a mouse model? Yeah, so I mentioned at the beginning, cells can produce sialic acid from the glycolysis cycle by a specific enzyme, and there is an enzyme which have a very complicated name, which is called UDPs, N-acetylglucosamine eparase, N-acetyl monosaminase kinase. I always call it GNE because it's a too complicated name, but this enzyme specifically is important and essential for the sialic acid production, which then goes to this nucleus. There is another enzyme, there's another knockout meanwhile available, the CMAS enzyme, which also can be used to study this. So if you knock out sialic acid completely in mice, it's lethal. So the mice die early in embryonic development, about E12, E14, and they have then overshooting complement activation. Yeah, so they have a very strong complement activation and cannot go into development, in full development to this complement cascade. Therefore, we analyzed heterozygotes. Yeah, so meaning that they have an intact enzyme on one allele and the other allele is deleted. Most of these heterozygous mice have no phenotype at all. So normally mice which are heterozygous compensated by the one allele which is still available. But apparently sialation production, at least by this enzyme, cannot be fully compensated. And therefore they have a little bit reduced sialic acid on the membranes. And this mimics very much the aging situation. So if we look in aged brain, in an aged erythrocyte, in aged organs, we normally have a little bit reduced sialic acid from the age of 80 onwards. Yeah, so there we normally have reduced sialation. So we mimic this in a mouse model, and then we first confirmed, yes, the mice have less sialic acid. So we stain polysialic acid with an antibody. Here in gray, you see the intact situation. You see it's going up. And even in an intact mouse, it's going down if the mouse is aging, the polysialic acid. In a heterozygous, which have this reduced sialic acid, it's staying reduced all over the time. Apparently, we have some compensation if it's going up in the wild type. So there we have the A2B5 staining, which is a ganglioside. So there we have a trisialic acid on a ganglioside. Apparently, this is even going up a little bit in aging. This is the immunostaining, and we quantified it. Yes, polysialic acid is going down in a mouse, an adult 9-month-old mouse, if we have deleted one allele of this enzyme, which is producing sialic acid. So we also look now in the central nervous system, and this is now a staining for neurons, which are labeled here in green by tyrosine hydroxylase. A molecule which is involved in the dopaminergic development and dopamine production of neurons, and a marker for neural cell bodies, NeuN. And then we quantify the number of these TH-positive neurons in the substantia nigra pars compacta. So we selected this area because there it's possible to quantify exactly the amount of neurons which we have there in this region. And what we saw is, here is the normal situation labeled in gray. And then we have in black the heterozygous, which have a reduced sialic acid. So we saw an premature, an early loss of the neurons. So at 6 months, we have a dramatic loss or a significant loss of the TH-positive neurons in the substantia nigra. And we also stained it for TH and quantified it for NeuN. So there was a premature loss of neurons, a neurodegeneration triggered by this reduced sialic acid in these mice. We also looked in another region, and this is the hippocampus. And in the hippocampus, you can quantify again the relative cell density in a region which is called CL3. And this is the wild type situation. This is the heterozygous situation. And there you see, quantification was already a challenge. The wild type situation, also there we lose over time neurons, not only in the mice, also on humans. But in the heterozygous, where we have reduced sialic acid, the loss was even more visible, even at an earlier time point. Yeah, so telling us also reduced sialation in the hippocampus, leads to a higher number of neurons which are lost during aging. So we looked also in the synapses, and in the synapses, there was even at 6 months, there was a dramatic loss of the synapses. So for the synapses, this phenotype is even apparently more visible. So apparently silyation prevents mainly also the loss of the synapses. And this is a double stabiling for PSD95, postsynaptic density protein 95, which is a postsynaptic marker, in VGLUT1, which is a presynaptic marker. And then you look for the co-localization of both puncta, which is then identified as a labeling for the synapses, And this is lost dramatically in the mice which have a reduced sialic acid, even at 6 months of age. We also analyzed the microglial processes. There is a microglial staining by IVO1. Then there is an imaging analysis where we reconstruct the skeleton by a software. And then we can see, yes, if we have a heterozygous mouse which have reduced sialation, the tree length. So the tree length is the total length of these processes which the microglia have is reduced. The branch number is reduced. The number of junctions and the tips are also partially reduced. So telling us also that the arborization of the microglia is changed. It's reduced if we have a little bit less sialic acid in the mouse. So we had a phenotype. If we have reduced sialation, we have neurodegeneration, we have a little bit microglial changes. If we summarize this, the gene E mice with reduced sialation shows increased age-related loss of neurons, synapse loss in the hippocampus, but no typical inflammatory gene transcript profile, and reduced microglial arborization. So the story was not at the end for us. So we were asking, yes, our in vitro data showed that complement is involved in removal of synapses and neurons. So we were asking, could we mimic this also in the mice? Yeah. So we were taking these mice which have reduced sialation and crossed it with a mouse where we have no alternative or no complement activation at all. If you knock out the complement factor C3, all the different complement pathway merge. So in principle, you shut down complement activation in these mice almost completely. So what is happening if we now knock out complement factor 3? I can make it short. So we can completely revert the effect. So I mentioned before, if we have reduced sialation, we have less neurons in the substantia nigra if the mouse are becoming older, 12 months, for example, old. But if we now knocking out the complement cascade, the C3, we have reverted the number of neurons. Yeah? So there is no loss of neurons. So prevented the neurodegeneration by knocking out the complement factor C3. And this is shown here as a quantification, reduced new and positive neurons in 12 months. If we have reduced sialation and the complement C3 deficient mouse, independent of the gene E phenotype or genotype, is reverted. Yeah? And the same is with the tyrosine hydroxylase positive dopaminergic neurons. So this is a marker for the dopaminergic neurons. The same was the case for the hippocampus. Here we had to quantify quite a lot because the effect was only minimal, but again, it was completely reverted. Yeah? So reduced sialation, as we have in aging, is leading to less neurons in the hippocampus CF3 region, and knocking out the complement factor C3 is reverting the loss of these neurons, the neurodegeneration. And the same is with the synapses. So here I only show the complement C3 are knocked out. It's completely reverting the loss of the synapses in these mice. And also the microglial phenotype. Yeah? So this reduced arborization of the mice was reverted if we have no complement factor C3. So this is here the wild type situation. Here is the gene E heterozygous in black. And then we have here the complement knockout. So it's coming back again, the tree length, the branch number, the junctions, and the tip. Yeah? So telling us, yes, there is an involvement of complement factor C3 in the whole neurodegeneration after reduced sialylation. So what can we learn about this? If we have G&E heterozygous mice which have reduced sialylation, and if we now completely knock out the complement cascade in a C3 knockout mice, we show a rescue of the age-related loss of the neurons. So we can prevent neurodegeneration by removing the complement cascade in the mice. We can rescue the loss of the synapses in these mice if we have no complement cascade. We can rescue the inflammatory gene transcription of these mice, and we can rescue also the reduced microglial aberration. So apparently we can completely revert the effect of the reduced sialylation in this mice. So we have told you a lot about the mechanism, complement, about Siglec receptors which recognize sialic acid. Then I showed you what is happening if we mimic the aging situation in a mouse by reducing sialic acid. I explained to you that this loss of the neurodegeneration, loss of the neurons is mediated by the complement cascade. So now we are coming to a disease, a human disease, which is called age-related macular degeneration. And we developed a therapy approach for this disease. And finally, I will show you that this is now also going into a clinical development, and that it's in the US in a phase 2 clinical trial for this age-related macular degeneration. So what is age-related macular degeneration? It's a disease which is mainly affecting the industrialized countries. About 15 to 20% of the population, which is 80 years or older, have this, and it's a very frequent loss of vision in our aging population in the industrialized countries. And the mechanism is closely related to complement, and it's closely related to microglia. So what do you have in this disease? You have an overshooting complement cascade in this retina, and this leads to degeneration of the retina. And we know that, for example, there's a close genetic linkage to complement factors. And second, we have a microglial activation, macrophage invasion, and they also contributes to inflammation and oxidative burst and oxidative damage in this disease. So for us, it was an ideal target to test whether our concept and mechanism is really true in this disease process. So we were taking defined polysialic acid. We were taking polysialic acid, and this is shown here with a degree of polymerization of 20. I showed you there are monosialic acid, trisialic acid. Here we have 6-sialic acid, and it's going very long, the long-chain polysialic acid. But we decided to take this monomer because we had some pre-experiments where we analyzed that this is optimal for Siglec stimulation and have almost no toxicity. It's binding to Siglec-11 receptors, which we have in humans on microglia, also in the retina. It's also binding to other Siglec receptors. And I showed you before, it's inhibiting the complement activator property. Yeah? So it's doing both. It's targeting the complement cascade as well as the microglia, the Siglec receptor on the microglia. And therefore, we think it's an ideal therapy for age-related macular degeneration, because both pathways are instrumental for the disease process. It's human identical if it's produced by bacteria. It's biodegradable, it's an anionic molecule. And clearly we have to use humanized mice to mimic, to have an animal model. Yeah? So we first generated humanized Siglec-11 transgenic mice, so putting this human Siglec-11, which the mouse normally don't have, into the mice by an artificial IBA1 promoter. So getting the Siglec-11 on the microglia and the macrophages. Since mice have no equivalent polysialation recognizing Siglec receptors. We later realized also Siglec-E is able to recognize a little bit polysialic acid, but the sensitivity and the effect is 10 times less. So you need a 10 times higher concentration to activate the Siglec-E receptor of the mice compared to the Siglec-11 receptor in humans by this polysialic acid. So this is now the setup. So we were taking these mice. We first did a laser lesion, yeah, and then 1 hour later, 1 hour later, we injected polysialic acid into the eye, into the vitreous body. And this is in normal therapy in humans. Yeah. So also in humans, very often, if you have a wet form of the age-related macular degeneration, there is an antibody against vascular endothelial growth factor injected locally in the vitreous body. So this is not dangerous and a very routine in the ophthalmology departments here at the clinical units. So we did it in the mouse. We had a wild-type mouse. We had a humanized CYCLAC11 transgenic mice. We injected a control. We injected 2 different concentrations, 0.2 microgram, which is quite a lot. It's only 200 nanogram of this substance. And we injected a higher concentration, which was 3 microgram polysialic acid. Then we had this laser lesion, and 2 days later, we analyzed the mice in respect to microglia activation, macrophage activation, vascular leakage, which is also a typical scene in this age-related macular degeneration in humans, and complement activation. This is a very artificial model. Yeah, clearly age-related macular degeneration in humans is not a laser lesion, but it's an accepted animal model which many people are using. Yeah? So in principle, we make a very severe immune challenge lesion by this laser into the retina of this mice, but it's very often used as an animal model of age-related macular degeneration. Here are now the data. So we imaged the retina by taking out, staining them with IBA1, and then you can say you have the wild-type situation. If we make a laser lesion here, it's a plus. You have a lot of microglia activated in this laser lesion, the microglia. You also get some microglial activation in the humanized CYCLAC11 transgenic mice. It looked already a little bit less, and it was already a little bit less due to this additional inhibitory receptor in this mice. And then we injected the 2 different concentrations of this polysialic acid. The low concentration was able to act in the humanized CYCLAC11 transgenic mice. So there, even at 200 nanogram and 200 nanogram dose, which we injected into the eye, we were completely shutting off this microglial activation in the retina. In the higher concentration, we also achieved this effect in the wild-type mice. And I mentioned they have also another receptor, CYCLAC-E, and apparently if you have much higher concentration, you can also target this receptor. And there's the quantification. Yes, wild-type CYCLAC11. If we treat them with polysialic acid, we reduce the microglial activation, higher concentration in the wild type, and even a lower concentration in the CYCLAC11 transgenic. And we also analyzed the choroid, yeah, so the macrophages which are invading into the retina, and there we saw the same effect. We also analyzed the vascular system. And therefore, we did fluorescence angiography, which is also done in humans. So if you have age-related macular degeneration, very often you can do fluorescence angiography. So you inject FITS labeled dextran, and then you can see the leakage of the vessels. So these are the intact vessels, and there's almost no leakage after a lesion, but there's leakage after lesion, which you can see here in the 3 laser spots, which I've done. And if you try and treat with polysialic acid, there is almost no leakage visible. And this is also here shown in this quantification. So we also can inhibit the leakage of the vessels after polysialic acid treatment in these mice. And we also looked for the complement cascade. So here we use the staining of the terminal complex, of the membrane attack complex. At the beginning, I showed an ELISA assay where they analyzed this terminal complex formation. Here it's a staining. Then it's called membrane attack complex formation, which we use the staining for C5b9. And you can see here there is a complement deposition after the laser lesion in the wild type and CYCLAC11. And if you now give polysialic acid, it's turned down. And this turning down of this complement cascade is independent of the CYCLAC11 receptor. So in the CYCLAC11 transgenic, it was the same effect size as in the wild type for the low concentration as well as of the higher concentration. So this, I think, is now my last slide in the summary. So we tested an interval application of polysialic acid with a degree of polymerization of 20 in a retinal laser lesion model. We showed that this polysialic acid inhibits the microglia macrophage activation in the wild type, but also in the CYGLAC11. In the CYGLAC11, it was even acting at a lower dose. It reduced the vascular damage, both in the wild type and the CYGLAC11 transgenic. And in the CYGLAC11, a lower dose was even able to do it. And it inhibited the complement membrane attack complex. So the whole complement cascade was inhibited in this mice by both doses, in the wild type and the CYGLAC11 transgenic. So I mentioned there is now a therapy in development, and they were taking a very similar approach. They were taking polysialic acid. They added it to a backbone, so a lactic polyglycolic acid backbone. This had only the reason that they can file a new patent. By doing this, they filed a new patent, and then they got the money, and the money is available in the US apparently for these new approaches, and they put it into a phase 2 clinical trial by a new startup, which is called Avicida Therapeutics, where they wanted to generate a new treatment for dry age-related macular degeneration. So at the end, I wanted to thank my team, particularly the ophthalmology unit, University Hospital of Cologne, where we are closely collaborating also with the complement unit in SCACA stream, also with the Hanover teams which we got funding. And then we have also funding here from the Excellence Cluster in Germany and the German Science Foundation, as well as from European projects like the FAGO project. In particular, I wanted to thank all the members of my team, which were doing the work and which pushed all the research forward. Yeah, thanks a lot. I think now I'm happy to take questions.
Thank you, Professor Harold, for this nice presentation. I really enjoyed it, and it was very nice. We learned a lot from your speech, and especially about the salicylic acid and how the clinical trials are conducted for polysalicylic acid as for determination of some eye disease. So I would leave now the stage for questions. Is there any questions? Please raise your hand. Okay, we have one question, two questions here for Dr. Shahira. You can make your onto your mic. Shahira.
I cannot hear you.
Could you make your mic unmute?
Yeah,
can you hear me now?
Now we can hear you. Yeah, now it's okay.
Okay. Good evening, and Professor Neumann, I'm very glad to have heard your talk. I had two questions. So first, you talk about the effect it had on the hippocampus and on the substantia nigra. Was it the same effect here and there? Was it the same significance, or was it the same effect? To the same degree.
Yeah, it was in both organs. It has the same effect. Silyation was reducing the number of neurons in the substantia. If you have reduced silyation, it's less neurons in the substantia nigra as well as in the hippocampus, and both was reverted in the complement C3 knockout.
Okay, so would you then expect that not only Alzheimer's disease would be affected, but also Parkinson and Huntington?
I think so. So I mentioned you also in my talk that I don't believe fully in this linkage to Alzheimer's disease. In my opinion, you cannot clinically, or it's so far not done clinically to make a distinction between Alzheimer's disease and dementia. So for me, this sialic acid, cyclic axis is mainly preventing aging, and any oxidative, any inflammatory burst and dementia effects during aging.
Okay, so I think that was it. Yeah, that was my two questions. Thank you very much.
Thank you, Dr. Ashira. There is any other question? Okay, can I have two questions, please, Professor Howard? It's one about the your talk explain how the influenza virus, when it attacks the mucosal membrane, it take out the salicylic acid, so it increase the immune response, innate immune response against the influenza virus.
So you are not talking about infectious diseases, which is not really my topic.
Yeah, but it might explain why the immune system is triggered when the influenza virus attack mucosa membrane.
The virus use the sialic acid normally as an entry in our body. So they have specific receptors to bind to sialic acid, and by this they can enter in the cell, and then they want it to be released from the cell surface to a neighboring cell, and therefore they have neuramidases, so they have sialidases, so have again specific enzymes to bind to the cell, and then also to shed and to enter and go to a second cell. But the loss of sialic acid is not directly related to a viral infection. So in a viral infection, the body normally, as soon as the virus enters the cell, will recognize that there is a virus inside, and then they even will remove their own sialic acid by sialylation, by neuramidases, to activate the immune system. So my topic is more about sialylation in aging and preventing immune responses. If you talk about viral infection, you have a completely different situation where you need loss of sialic acid. Yeah, so the extreme situation is a septic shock in our body. We lose about 50% of the sialic acid on our everywhere on our cells if we have a septic shock in our body, because you need this immune response to fight against these bacteria.
Okay. And the second question is about if there is any comparative study between using polysilylic acid for as anti-complement, decrease the complement activity, and antibody against complement, like C5b or C3b.
No, so far these data about the inhibition of complement by polysialic acid are very recent, and it has not been tested anywhere in humans. What is known that the people were using in specific human diseases antibody against complement factors as a treatment, but all these studies failed because the humans had a higher susceptibility to certain bacterial infections. So apparently it's risky to completely shut off the complement system in humans. Yeah? So you become susceptible and you have a higher risk to get very severe bacterial infections. Whether this is the case also for polysialic acid, it's unclear, because antibodies cannot be turned off by our body. Polysialic acid can be turned off by our body by own neuramidases.
Okay, but you think that in clinical trials, the polysilylic acid will not have the same adverse effect as antibody for complement, against complement?
The problem is not given if you locally apply the polysialic acid in the eye. So the approved medicine for age-related macular degeneration, which was approved a few months ago, is an complement inhibitor C3. So the local inhibition of the complement in an organ like the eye does not really need to increase bacterial infections. It's only if you give it systemically, then you have a higher risk of bacterial infections.
Okay, thank you very much. Is there any other questions? Okay, thank you, Professor Hanoura, for your nice presentation, and I enjoy it. And I think okay, there is one more, Asma Ali. Okay, Asma Ali, you have a question? We can take.
Yes, doctor.
Can we take this question? Okay.
Okay. First, I would like to thank you, doctor, for this interesting topic. My question is, since sialic acid is present in breast milk, is there a chance to develop a milk formulation for babies with high risk or with already diagnosed irritable bowel syndromes or Crohn's diseases, since it can act as anti-inflammatory or shut the inflammation? Is there a chance to develop such?
This is a concept where people are working on. Yeah, so indeed, there are industry and also startups working on human milk oligosaccharides to improve bowel disease, inflammatory bowel disease. So this is a concept which is very valid and which is on the way. The problem, I've not gone in depth into the sialic acid. So the bovine milk which we drink is not optimal for humans because the other mammalian species have 2 types of sialic acid, while humans have only 1 type of sialic acid. Yeah? So if you make such therapy approaches, you have to pay attention for this, because if we eat red meat and if we drink bovine milk, we incorporate a wrong sialic acid in our body and we get chronic inflammation and chronic antibody responses in our body. So one have to be careful to really substitute as a supplement the correct form of sialic acid, not as monosialic acid, again as an oligosaccharide. If you give it as a pure sugar, monosialic acid, you will completely feed the bacteria and you will have a microbiota which is completely out of range.
Okay, thank you, doctor.
Thank you again, Professor Hanoura, for your presentation. And now I leave the stage to Professor Dr.haruald.
Well, I would like to thank you, Professor Neumann, for the interesting talk, and I think the engagement of the audience showed how interesting goals for everybody. And I would like also to thank Dr. Amr for the nice moderation and of the session. And I hope we can have you once more in our Let's Talk Science. And maybe once we can invite you to Cairo. We'll be happy to host you here. Thank you, everybody, for attending. And with this, we end our session today. And see you next month with another guest. Thank you. Bye-bye.
Thank you. Bye-bye.
Yeah, thanks a lot. Bye-bye.